Hi, my name is Didi Fabian, and I will present a study titled Revisiting Low Penetrance RB1 Gene Sequence Variants, which is a collaboration together with geneticists and bioinformatics from Tel Aviv University. This is a visual representation of the 3D structure of the RB protein, which is essential for its function. Here in high magnitude, its normal structure, and the sequence variant. Any change in the RB protein's DNA sequence can alter its folding and ultimately its functionality. Now, retinoblastoma is a model for high penetrant mutant RB1 alleles. Yet in some families, penetrance is incomplete with mutant alleles referred to as low penetrance. Now, a low penetrance gene refers to a gene in which variations or mutations have relatively small chance of leading to a particular disease, such as with this individual. Several mechanisms have been proposed for low penetrance in retinoblastoma, some more convincing than others. I won't go through the entire list. The main idea is that either insufficient protein is produced or the produced protein only partially functions, impacting its activity. The goal of this study was to review previously reported cases of low penetrance RB1 sequence variants and reevaluate their putative pathogenicity using advanced bioinformatics tools. We conducted a literature search using relevant terms, as you can see here, but it's important to note that this was neither a meta-analysis nor a systematic review. We included papers that provided sufficient genetic information about the mutation and only those with asymptomatic carriers. Now for the bioinformatics part, which is often a black box for many. We use large-scale databases and algorithms that translate genetic sequences into protein products. The outcome measures were cases reported to be originally in the original papers to be low penetrance and downgraded using our tools to a likely benign sequence variant. Specifically, we used Ensemble and Alpha missense software, and those cases that were downgraded were further analyzed using the American College of Medical Genetics and genomics guidelines, which you can refer to in the literature. Now for the results. Altogether, we found 386 papers, of which 44 had sufficient genetic data, and 32 included asymptomatic carriers. Of these, 7 sequence variants were downgraded to likely benign, and of these 5 met the ACMG criteria. And here are the details of the 5 mutations and the effect on the protein, and the list of papers where the mutations were reported. So to conclude, using bioinformatics tools, 5 cases previously assumed to be low penetrance RB were downgraded to likely benign RB. These so-called inherited RB cases are in fact sporadic RB that carry sequence variants in the RB1 gene, which we presume to be a rare polymorphism that is unrelated to the phenotype. There's no need for genetic counseling to any of these family members, and there's no need to offer prenatal diagnosis or surveillance schemes to asymptomatic sequence variants carriers. Thank you.